So when we observe the blood smear, how to identify the individual types of foreign blood elements there? Let's start with the most easy task, which are the erythrocytes or red blood cells, RBCs. They have a shape of a biconcave disc. So this would be the front view, this would be the lateral view. The central region is thinner, okay, that's why it appears also lighter and more pale in the center. The diameter is approximately 7.5 micrometers, depending on the fixation process. The thickness of the disc is approximately 2.5 micrometers, and the thinnest region in the center is 0.8 micrometers. The shape is a biconcave disc without nucleus. It's mostly filled with hemoglobin. Each red blood cell is supposed to contain between 28 to 32 picograms of hemoglobin, per each individual red blood cell. And the product, if we consider also the red blood cell count and the hematocrit, will be in male 130 to 160 grams of hemoglobin per 1 liter of full blood, and in female it's 120 to 160, these are the reference values, grams of hemoglobin per 1 liter of blood. Next foreign blood element would be thrombocytes. Or blood platelets. They have an irregular shape with many invaginations, but you can see that the center has a granular structure, so that's how you distinguish the peripheral clear hyalomere region and the central granulomere region. It contains granules with many active substances that could be released once the thrombocyte will be activated during the hemostasis, during the blood coagulation process. And these substances are, for example, calcium ions, ADP, adenosine disphosphate, ATP, adenosine trisphosphate, platelet-derived growth factor, von Willebrand factor, serotonin, and other substances involved in blood coagulation. The size ranges between 2 to 4 micrometers, so these are the smallest elements in the peripheral blood. Now let's discuss the granulocytes. Let's start with the neutrophils. The neutrophils are round in shape. They have a segmented nucleus consisting of several lobes connected by thin chromatin bridges, and in the cytoplasm you can find fine granules, neutrophilic granules. They are neither basophilic nor eosinophilic, they are neutrophilic. Neuter means in Latin neither of two. So the size of the cell is like 10 to 12 micrometers, and we have this segmented nucleus. And the fine or small neutrophilic granules. These granules contain bactericidal enzymes that can kill bacteria. The cells work as macrophages. They are able to perform phagocytosis. The segmented nucleus may have 1 to 5 lobes, and there is a relation between the age of the cells and the number of the nuclei. So if you have the number of lobes and you will order these from the youngest forms with only 1 or 2 lobes to the oldest forms with 5 lobes, then the average number in a healthy individual is somewhere around 2.7, which is also known as the nuclear number. And in patients where you will find shift to lower average number of lobes, it's called shift to the left or left shift. It means the younger forms prevail. If you will find in average more often highly segmented nuclei in neutrophils, it's called right shift. And this will inform you about the average age, because young cells have one nuclear segment, and elderly cells have 5. Why is this relevant? Think about a typical situation where more young cells are entering the peripheral blood from the reserves. So they could be provoked by bacterial infections, by bacterial toxins. So that's where you observe live shift typically. The right shift could be observed in situations where the formation of neutrophils is somehow blocked or impaired. So you have older population in the peripheral blood. This is also called, this order from the young to the older forms is also called the Arnett's count. The youngest forms that have just entered the peripheral blood with only one segment, so these are also called rods or step cells. And these various shapes of the nucleus gave also these cells the name polymorphonuclears. Okay, the next type of the foreign blood elements would be the eosinophils, the eosinophilic granulocytes. They also have segmented nucleus, but they have 2 lobes. Okay? And the granules are there, but much larger, and they are eosinophilic, so they are really orange to reddish, much more apparent. So we have a bilobed nucleus. We do have large eosinophilic granules. That contain cytotoxic and antimicrobial and anti-parasitic substances, such as major basic protein, eosinophilic cationic protein, and other cytokines and pro-inflammatory molecules. The size ranges between 10 to 15 micrometers. Okay, our next type would be the basophils. Basophilic granulocytes. These cells have an irregular shaped nucleus, which also has lobes, but irregular shape. And the cytoplasm is filled with large basophilic granules. The nucleus could be hiding partially behind these granules because they stain very intense, in a very intense way, and they are dark purple to dark blue. So we got the irregular shaped nucleus and the huge basophilic granules that contain substances such as histamine and heparin, which somehow resembles the content of the granules of mast cells we have seen in connective tissues. There are also leukotrienes and inflammatory cytokines. The size ranges between 8 to 10 micrometers, so these are typically the smallest granulocytes. So we got these three types of granulocytes, okay? Next we got the agranulocytes. First let us discuss the lymphocytes. And the lymphocytes, they have typically a round-shaped nucleus. They could be small and they could be large and they could be medium-sized depending on their activity. But they always have a round-shaped nucleus which fills most of the cell, and the cytoplasm, which is very limited, especially in the small lymphocytes, contains only so-called eosinophilic granules, which are lysosomes. You can see these granules only if you really try to use your fine focus knob in the microscope and you have a high resolution oil immersion objective. So this would be a small lymphocyte. This would be a large activated lymphocyte. The size will range between 6 to 12 micrometers, so you can have small, medium-sized, or large lymphocytes. Lymphocytes represent a highly heterogeneous population, so no wonder you can have you can have a variable morphology. Then we discuss the monocytes, which are actually the largest cells of the peripheral blood. The nucleus is kidney-shaped. And in the cytoplasm you can find only delicate azurophilic granules. So the size is 15 to 20 micrometers. The largest cells in the peripheral blood, we have the kidney-shaped nucleus, the azurophilic granules, which are lysosomes. And as we already know, the monocytes migrate from the blood into the tissues, so they are leaving the bloodstream, which is called extravasation, and by entering the tissues they mature into macrophages. Lymphocytes have only few cytoplasm, or little cytoplasm. So what's important when you observing foreign blood elements in a blood smear? Why don't you focus on the morphology of the nucleus? Ask yourselves if it's present, if it's segmented, if so, how many segments are there, if it has some lobes, or if it's round-shaped or kidney-shaped. And the second feature, which will bring you to a reliable diagnosis, will be looking at the granules. Are they present? How do they stain? What's the size? So by combining the morphology of nucleus and the morphology of the granules, you can classify these foreign blood elements quite easily. Good luck!